So let's go. So let's start with the basics. Okay. Maybe you remember this Hardy-Weinberg equilibrium. That's what we call in French la palissade, which means basically saying that nothing's changed if nothing's changed. And what this is about is simply if you have some requirements that are fulfilled, like there is no selection, no mutation, no migration, and a large enough population and random mating, if these are fulfilled, then allele frequencies will not change in the population. They will remain stable. Of course, this is a very artificial model, and no population probably never fits this model, but this is precisely what we are interested in. We are interested to know when allele frequencies change in a population, why they do so. And they probably do so because one of these 5 requirements are not fulfilled. And of course, well, mostly what we are interested in here in adaptomics is to find if selection is involved. Okay, and before we move on with selection, we first need to be aware of another paradigm, another null hypothesis, which is called the neutral theory paradigm. You probably also heard about it, and the idea is that most of the variation we see at the molecular level does not affect fitness. It's completely silent, let's say, when we talk about fitness. So it's not due to selection, and instead the genetic variation we see is best explained by stochastic effects or demographic processes. And again, here what we're going to be interested in is to detect selection, and to do so it means that we need to test whether the allele frequencies we have simply match the neutral evolution paradigm, or if it deviates from it. So of course we're going to need to be aware of demographic processes and stochastic effects. But just for you to know, like, population genomics might sound like very crazy things to do and crazy elaborate. So certainly it is in terms of models applied, but the type of data you work with when you do population genetics is simple and only allele frequencies. You generate allele frequencies, and from that you're going to be able to tell a lot. You're going to be able to tell a lot about the life history of a population, if there is specific selection acting at a specific locus, if there is some negative interactions between distant locus in the genomes, and these kind of things. So simply with just allele frequencies. Of course, now the cool thing is we have the genomics tools, which means that for basically virtually every single nucleotide we can assess allele frequency, and then for every single allele nucleotide, we can know whether this nucleotide fits the general neutral evolution pattern or if it deviates. And from that we can move on, and then you can focus on genes, whatever transposable elements, whatever is interesting, then you can just go for selection or finding or testing for this null hypothesis. So yes, so a lot of possibilities. But as I said, we first need to be aware of demography and how it affects genetic diversity. So the lecture will be separated in several short videos simply because it's easier to digest. So we're going to start with this one, and for next chapters there will be a new video. So, demography. This is probably the most famous case of drastic demographic change, and which is a bottleneck. And the bottleneck, what it is simply you have reduction of the population size and the effective population size. I will come back to this, which then leads to diminution of individuals, and then eventually either extinction or recovery. And a specific case of bottleneck is the founder effect, which is from a large population, then few individuals will move to or colonize a new place. In this case, but in this case as well, when it's about like a catastrophe, for example, like coronavirus, the thing is this will create, or this will reduce genetic diversity. In the case of founder effect, simply because the individuals moving here will not be able to carry the total range of genetic variation of this huge population. So for sure there will be loss of genetic diversity, but also increase of homozygosity, simply because a few individuals will be probably related to each other, and then mating will lead to homozygosity or increased homozygosity. And last but not least, there will be loss of rare alleles just by stochasticity. Like this one, the probability that this rare allele is carried by one of these founders here is quite low, so it's gonna get just removed. And well, it can be catastrophic, can be a founder effect, but something quite important in plants is transition to selfing. So in Brassicaceae and in many species, plant species, outcrossing happens via the self-incompatibility. So this species, Capsella grandiflora, is not able to self, basically, because there is some mechanism, some molecular mechanism preventing it. But two of the species actually lost this ability of self-incompatibility, which means they became selfers. They became selfers, and with that you can imagine that because few individuals will carry the mutation responsible for loss of self-incompatibility, it means that the pool of genetic diversity will be super low to start with. And also because they will self with themselves, then homozygosity will dramatically increase across generations. Okay, so I mentioned it just before, there is something important to consider is the effective population size. It's quite important because, so what we mean by effective population size is the size of an ideal population, for example with low inbreeding, that would give rise to the same genetic features like number of alleles, number of rare alleles or their frequencies as the real population that we are interested in. So for example here, you have two things, the actual real population size, which is stable across generations, whatever, and the effective population size. And this is put into relationship with the sex ratio. So what you see is that with a distorted sex ratio, the effective population size strongly decreases. So this is from male to female, but in the direction will be exactly the same thing. So what it is about and why it's like that, simply because here you're gonna have, let's say, one male for 100 females, which means that all the progeny will inherit from only one father. So you understand that then with only one father for all the next population, that means that genetic diversity will strongly decrease, and it will decrease because only one individual gave rise to the new population. So the effective population size is simply 1. Even though there is hundreds of individuals, they all carry the genome of one and only single individual. Okay? And this is a problem. This is a problem notably for breeding. So like breeders have developed the habit to have what they call elite males when it comes to cattle, which means that they will have the sperm of one elite male and fertilize hundreds and hundreds of females with the sperm of only one elite male. Because of course they want specific traits, whatever. But the problem with that is exactly this scenario here. So we are rather around here, which means very low population, effective population size, which means low genetic diversity, which means strong inbreeding, which means deleterious mutations, which means very low resistance to pathogens, for example. And when it comes to humans, we are billions on Earth, but actually the effective population size of humans is around 10,000. Yeah, 10,000. Which again is exactly the same scenario here, more or less, is that with quite a small population to start with, like in human history, then humans colonize the whole Earth. And it's precisely a founder effect again, like let's say from this original 10,000 population, human spreads, but of course the genetic diversity in humans is pretty low. So, if we continue on this idea, how effective population size will affect selection. So imagine a case of, let's say, close to our divine bag situation with large population, palmitic population, etc., etc. But there are going to be though something which will deviate from our Van Berg equilibrium, which is having selection, and there will be an advantageous allele for this specific environment, let's say, and deleterious one. So in this case, actually, they're going to be quite a strong opportunity for selection to act, and stochasticity, which means again change of alleles just to drift, just to random events, will be quite low, because that's precisely the point of Hardy-Van Berg equilibrium. Like if population again is large enough, etc., then the chance of having an allele fixed or disappearing by stochasticity is very low. So it means like allele frequency changes have a strong probability to occur via selection. And indeed, we can imagine that this allele is advantageous and will have a chance to spread across the generation, across the population. Sorry. But now let's imagine another scenario. They're going to be founder effect, so like a small population with two individuals, one carrying the advantageous, one carrying the deleterious allele, will then move. In this case, we are in the opposite scenario. We are in the scenario where stochasticity will play a strong role, simply because if one of the two individuals die, it means that the population will have fixed allele or removed allele. So simply, if this one died, this one will be fixed. If this one survived and this one died, then this allele will be fixed. So stochasticity will play a strong role, independently whether this allele is deleterious and this one is advantageous. And it's precisely the case. If this one dies for whatever reason, falling from a cliff, then all the others, or the next generation or the next population, will only carry this advantageous allele. Well, if it's this one dying, then the population will carry only deleterious alleles, simply due to stochasticity. And take-home message here is simply, with a low effective population size, there's going to be a low efficacy of selection. Selection will not be able to act or to counteract stochasticity, let's say. Okay. So, next point. Now let's change a bit in the same idea, but yet a bit different. So imagine you have several individuals in the class here, and each of them have three loci here in a given chromosome, A, B, and C. And each of them have certain variety of alleles between them. So what you can see here is that, for example, when you have A prime here, you always have B prime allele there. And same here, A blank, B blank, A double prime, B double prime, A plus, B plus. So what does it say? That says that basically if you have a certain allele here, you always will find this certain allele there. And that's what we called linkage. That's what we called genetic linkage. That's what linkage disequilibrium is about. Is basically, it translates the idea that two loci will always segregate together when it comes to recombination during meiosis. And because they will always segregate together, simply because there is no recombination hotspot between them, it means that finding one allele here will automatically means finding associated allele there. And now imagine the third locus here, C, because you have plenty of recombination spots in between, then there is no chance that we can predict the allele in C when we know allele A or allele B. And as you can see, for example, C double prime can be found with this combination or this combination with no difference, and other alleles can be found even though here we have a certain type of allele. So that says that basically the C allele is completely unlinked to A and to B, because precisely there is recombination happening in between them. So that's what I'm saying here. So we say that there is linkage disequilibrium between A and B, simply because, again, they will always segregate together during recombination. There will never be recombination between them. While it's the opposite with C, C will most of the time be completely unassociated with alleles A and B there. Okay. And another important thing is that linkage disequilibrium is a marker for what we call effective recombination. So effective recombination depends on two things. It depends on the recombination itself, of course, but also on genetic diversity again. So now let's consider 4 other individuals from the class. Now imagine that actually all of them carry A prime, B prime, and C plus there. So what we're gonna see there is basically we could assume that C plus allele is always associated with A prime and B prime allele. And that means that basically here what we can say is that A, B, and C are very linked. There is a strong linkage disequilibrium between A, B, and C. And not because there is no recombination, it's like before, there is the same amount of recombination between them, but because there is no genetic diversity, virtually, recombination happens physically but doesn't change anything in the combination of alleles. And that's precisely what we called effective recombination. And here, effective recombination is actually very low or completely inexistent. And that's the case here. So for example, so that's how we represent usually linkage disequilibrium. So usually linkage disequilibrium, because of recombination, decrease with the distance between two locus. So if A and B, they're probably somewhere around here, and A and C are probably somewhere around there. So A and C usually would have a linkage which is very low, while A and B would rather have a linkage which is super high. So they always find together. And this is expressed here as just a correlation between alleles. It can be expressed as different values, but R2 is one of them. So you have here the comparison between cultivated and wild mung bean, for example. So what we can see here, and it relates to this idea of effective recombination again. So recombination itself between these two species is probably very similar in terms of processes, like where recombination happens. But nevertheless, you can see that linkage disequilibrium in the wild decreased very strongly, very fast. Like basically after 100 kb, like two loci will not be linked anymore. There will be completely random segregation between them. While if you look at the cultivated one, you still find kind of a strong linkage disequilibrium between loci that are far further than 200 kb. And probably the difference between these two is precisely genetic diversity. It's well known when in crops that genetic diversity of crops is much lower compared to wild species. So what we can imagine here, what's happening here is precisely the case I described here, where genetic diversity is so low that basically these guys are linked, not because there is no recombination, but because there is just no genetic diversity or diversity of alleles to show this recombination process. And now just a word on recombination processes. It is well known that depending on the place of the chromosomes, then recombination rates varies a lot. And especially if we look at the centromeres, which are here, and here you have the recombination rates. Basically what you can see is like, same here, what you can see is that the recombination rate is usually very low around centromeres. And the further you go from centromeres, the more recombination you get. And in repeated regions, in general, that's what's happening. So now, just one more thing. So imagine now we're again in the same scenario as before, so very low genetic diversity. So very low effective recombination, okay? And now imagine that we have the case where A prime allele is actually advantageous, but B prime allele is deleterious, and same for C+. Here, selection will have problems to act, simply again because there is no effective recombination. It means that they will never be able to have a removal of deleterious alleles B prime and C+, without removing A prime, which is advantageous, and vice versa. There will never be the possibility for selecting A prime individually and getting rid of deleterious alleles B prime and C+. So basically that means that with low effective recombination, you're going to have low efficacy of selection. And you will have deleterious alleles that are always selected with advantageous allele. And that's it. And that's more or less what I said here. Okay, and as a summary, so it's a bit complex and we can just pause this video to read it through. This is more or less what I told you. So if there is bottleneck, founder effects, self-in transition, polyandry, anything that will then change or decrease the effective population size, that will have consequences on polymorphism, of course, and eventually on the adaptive potential. And why? If we go back here, so now there is increase of homozygosity also. There is a decrease of effective recombination. If there is decrease of effective recombination, there is increase in linkage disequilibrium, which means lower selection efficiency. You're going to have deleterious and advantageous alleles that will be linked together, segregate together, which means low selection efficiency, which means again a low adaptive potential of this specific population. And something which we'll talk about later, they're going to be also reduced purifying selection, which means removal of deleterious alleles, which means increasing what we call the genetic load, so the amount of deleterious mutations that are in a given genome. So let's summarize, and to summarize, I propose you a small quiz. So I ask you, according, and don't look to the video before, preferentially, if you want to test yourself properly. So according to the current paradigm, what drives most of the genetic diversity in nature? Is it selection, is it stochasticity, or is it demographic events? The next question is, if there is a bottleneck happening, it will decrease what? Will it decrease genetic diversity, or the efficacy of selection, or linkage disequilibrium, the efficiency of recombination or efficient recombination, homozygosity, the effective population size, or the stochasticity on allele frequency changes?